the University of Washington, and he's going to be talking about single-cell Hi-C data analysis.
Okay. Hello, everyone. Thank you for being here. And I feel it's really difficult to present another embedding work after these wonderful talks ahead of me. But our work is on projecting single-cell Hi-C data. So nowadays, we have a lot of single-cell high-throughput sequencing technology, like single-cell Hi-C to study the content organization, single-cell RNA-seq data to analyze the transcriptions, and the single-cell ATAC-seq for the content accessibility. So all these technologies provide a way to quantify cell-to-cell variability and also give a way to understand the underlying biological process. For example, if you have like embryonic stem cell and we have the single cell Hi-C data, you can imagine there are intrinsic dimensions. One of them will be the circular cell cycle, right? And there's also an orthogonal dimension that is corresponding to the linear differentiation. Therefore, we would like to expect a cylinder manifold out of these cells. So therefore, we formulate our problem also as a dimensionality reduction or manifold learning problem. So we are looking for a mapping function f, which map the original high-dimensional space, like the Hi-C matrices, to a lower dimensional manifold, like the 2D space with cell cycle time and differentiation time. For this audience, I would like to introduce a little bit of Hi-C matrices. So for each cell, we will have a contact map like this. So essentially, it measures how two locus on DNA are closed in 3D space. So we will have counts, integers in this matrix built into some resolution. If we have human data, like 3 billion base pairs, and we bin to 500 KB, we are essentially looking at this number of dimensions. So we are looking for some mapping from this higher dimensional space to a like 2D space. So we formulate as an unsupervised embedding approach, so we don't require any labels of the cell. And there's the steps from the single cell Hi-C contact maps. We first evaluate the similarity among them. So we will get a pairwise similarity matrix out of it, and then we will convert it to a pairwise distance matrix. Like we will get the Euclidean distance among the cells in terms of the Hi-C contact maps. And in the end, we will preserve the distance and we will project them into a lower dimensional space. I think you can expect that for distance preserving projection part here, we will use MDS. So this is a method if you are given a pairwise distance matrix among the entities, for example, 10 cities on the Earth, like we have the distance among them by air travel miles, and MDS will project them into a 2D space that preserve the distance, relative distance among the cities. Therefore, the whole procedure is complete, except we don't know how to evaluate the similarity among the single cells. So in the literature, in Hi-C analysis, people usually rely on this so-called contact distance profile. Essentially, from the cell, you can generate like a histogram. Essentially, it evaluates how frequent is contact in terms of their contact distance. So we will have contact distance profile from the cells, and we can calculate some measure like Jason-Shannon divergence, JSD, and use that as a pairwise distance. So this is the one way to evaluate the pairwise distance among the cells. So we got some single cell. There's one single cell Hi-C data available since last year from Nagano study. So this study studies the single cell the cell cycle effect, and they have the label for the single cells. And we select 120 cells, 30 of the cells are from each of the different stages of cell cycle, including G1, early S, mid S, and late G2. So we have this 120 cells, and we apply this procedure, get the contact distance profile, and we got a projection like this, different colors corresponding to different stages of cell cycle. You can see the cells are still separated, but not in like a circular pattern. Then we think about whether there's other method you can evaluate how single cell Hi-C data are similar to each other. One idea came across from this people looking at the reproducibility scores amongst bulk Hi-C data. So they're already, and we are thinking about whether we are able to use this method to evaluating reproducibility score as a similarity score in the single cell Hi-C data. There are already 4 methods developed to evaluating the bulk Hi-C reproducibility score, including HiCRep, GenomeDISCO, Hic-Spector, and QuasarRep. So they are slightly different in term formulation. Like HiCRep is for weighted correlation coefficient, and GenomeDISCO use some fusion map, and Hic-Spector, I believe, use some eigenvalue decomposition of Laplacian, and also Quasar is also based on Pearson correlation coefficient. At that time of our experiment, Quasar didn't work properly, so we only ran the first 3 on our data. So this is the result by applying Hic-Spector and GenomeDISCO together with MDS. It's still on the 120 cells from 4 stages of cell cycle. You can see the Hic-Spector together with MDS doesn't provide a good separation among the 4 stages of cells. But GenomeDISCO provides some separation, but there's no circular pattern. And we are surprised that HiCRep provide really good separation among the cell, as well as a circular order among them. Like G1 is here, followed by early S, mid S, and late S, G2. So there's a circular pattern out of it. So we are quite excited about this work. And then to remind you that HiCRep calculates correlation like this. So if you are given 2 Hi-C content map, the first step is it applies some smoothing among the locus. So it will make this remove the sparsity, and then it will control the contact distance and calculate the correlation. So if you are familiar with Hi-C content map, essentially you are looking at straps of them from the both of the maps and get a correlation score, and then you do the weighted sum. So this is a way to control the contact distance, but give you a way to evaluate how they are similar. I believe this is also helpful to recover like tag structure out of it. Then our method run like this from this single cell Hi-C contact maps. We apply HiCRep and calculate the similarity score, got this pairwise similarity matrix, and then we convert to this pairwise distance, and then we apply MDS. And then we apply them to this big 1,000 mouse embryonic stem cell, and we got the projection like this. And then we check the label of cell cycle of them. So they are essentially really come from a cell cycle. This is G1 phase, followed by early S, mid S, and G2, and they really show great separation among them. So we also think about how can we quantify the projection quality using some metrics. my mentor Bill Noble came up with this, I think it's a great idea, called the circular RC curve. So original RC analysis, you have the two class, and you can get a value from 0, 1, and you rank them and get the RC curve. But in our situation, there are two challenges. First, I think the easy one is we have 4 classes rather than 2 classes. Another difficulty is original RC analysis cannot handle the circular nature. So we got a circle, so we don't have a measure from 0 to 1, but we have this circular here. So Bill designed this measure like this. We first identify, for example, if we are only looking at early edge phase, like the red dot here. So we first identify the center of this class, so like here, and then we will calculate the distance in terms of angle, the angle, the cells away from how far away its cell from this center. So using that as like the 0 to 1 measure, and we can calculate RC score. And then we average across the 4 classes. So we got this average circular RC curve. For this, taking this as example, the area under the RC curve is 0.966, and this is the RC curve for early edge phase. So this is still our projection, and the original Nagano study in Nature also provides some cell cycle phasing. They use the percentage of long-range contact and also replication timing information, and they also place the cell on the circle, but our projection doesn't require that. And we calculate the area under the circular RC score, and the score increased from 0.911 to 0.936. So we show that by calculating the similarity in this principled way, we are able to provide a more accurate cell cycle projection. So another thing we checked is there are different single-cell sequencing protocols, like another single-cell protocol, Hi-C protocol is called sciHi-C, designed at University of Washington. It's essentially doesn't require you isolate the cell physically, but use multiple rounds of combinatorial indexing. So in this way, each cell can be tagged with a unique combination of labels. This sequencing protocol is very powerful, can give you more cells, but will produce shallower coverage. So therefore we are thinking about if we apply our embedding approach on other protocol, especially the coverage is lower, can we still provide a really good embedding? Therefore, we want to check how well the method works if we perform some downsampling. So this is still the same data set, but we only use a proportion of it. So this is roughly 20% of the cell, and the median coverage, the contact number is 88,000. And then we perform downsampling to 10K, 5K. It seems the cell cycle, the projection is still there. You still have a clear cell cycle pattern there. But once we move even further to 2,000 contact or 1,000 contact, they started to mix together. And this is the area under this is the average circular RC score from original coverage and downsample to 10K, 5K, 2K, and 1K. You can see how bad it can get in this once we do this downsampling. But it's still very good if you have 5,000 contacts. Another thing we check is if we have the shallow sequencing, can we jointly project with high coverage cells? Can we can save or rescue some lower coverage cells? So this is still the cells downsampled to 1K context, and we jointly project them together with original high coverage cells. And you can see after this joint projection, it can further separate them in this projection, meaning that if you have some shallow sequencing data, you can use a better, higher coverage cell to rescue the lower coverage. And the area under the RC curve increased from 0.71 to 0.883. Our method also supports joint projection of multiple cell type, probably not multiple species. For example, we joint project together with Nagano mouse embryonic cell together with another single cell HiC dataset available from another Nature paper is for oocyte-to-zygote transition. And this is the projection of the two dataset. The original, on the right hand part of this plot is from the Nagano study, and left part is from this Flamer oocyte-to-zygote transition cells. You can see the cell cycle still there, and the oocyte-to-zygote dataset on this side. And the oocyte resembled early as G1 phase. So they are connected here. So to sum up, this is the first embedding approach for single-cell HiC data. I'm quite excited about it, but I believe this is like the first step, rigorous computation for single-cell HiC data. It's more accurate and quite robust, and allows joint projection of multiple cell types. And I would like to acknowledge our NIH 4DN consortium as well as SCB Travel Fellowship Award. And that's it. Thank you.
Hi, this is really cool work. Thanks so much. I was just wondering if you have a sense of how many data sets you need as your input in order to get a reliable embedding, like your M value. Did you try downsampling that?
Okay, that's a good question. So we tried to 120. So you see it's already really good. I haven't tried smaller. I think initially I tried 20 with 5 from each category, and it's there. The cells, the circular pattern is there.
Oh, cool.
Okay, thank you.
So in terms of the lower dimensional space, you are projecting it into 2, k equals 2, or like how did you decide what was the lower dimensional space?
Yeah, the question is about how to decide the number of lower dimensional space. So some of the application you have some biological meaning, like the example I gave, you have the circular cell cycle time, that's one dimension, and differentiation, that's the second dimension. Sometimes you have this prior knowledge, the number of dimensions ahead of time. You can try that, but sometimes you can also like do some exploration of different numbers and see whether there's, I haven't tried this to like 3 or, I don't think you are able to visualize in 4. But I haven't tried 3. Okay. So usually I think it requires some exploratory analysis. I only use the input only input is the single cell HiC data. I didn't use any other information. Yeah, but it's possible to join with other type of sequencing data. We haven't tried that.
Very interesting work. I just want to ask, based on this figure, and if I want to know the cell type and the rank k-means, can I get the exact cluster as the cell type you show?
So I think the question is about the difference between our method and the k-means, right?
Yeah, so for example, I low dimensional, I embed the HiC data with your methods, and I get the low dimensional embedding and the rank k-means.
Okay.
Did you evaluate the accuracy?
Yeah, I think our method is more like dimensional reduction approach rather than a clustering. I think one key difference is you can imagine along this biological process, you don't necessarily have clusters, but it's a continuous spectrum.
I see.
Thank you.